Hello, everyone, and welcome. Today, we're going to be talking about T-cell activation. Now, really, this presentation is going to be putting together a lot of what we've already discussed in the past. So here, hopefully, the big intention is to consolidate and also just to maybe fill in some gaps. And you take as much time as you need, right? Now, T-cell activation, you're going to notice, is quite annoying, loss of better words. Well, not for the body, but perhaps for the learner. And this is because there's a lot of fail-safe mechanisms in place. You can be like, all right, what's the big deal? We need our T-cells to function. Why is it so hard? Why are there so many checkpoints along the way? Really, you can say because activating your T-cells is a big deal. What do you mean it's a big deal? Well, we discussed in the earlier, lecture. If you activate a CD4 T cell, helper T cell, that's basically a cytokine factory. That's going to direct your body, your physiology in a certain way. It's going to direct your immune system in a certain way. It's going to direct inflammation in a certain way. So if you have a cytokine factory in the works, if you're going to turn that on, it better be for a good reason. And similar rationale for CD8 T cells. They can directly kill infected cells. So if you have something that can kill other cells, it better be you better be sure that you're turning it on for the right reasons. So, and if there's a problem with any of these mechanisms, and you actually get autoimmune diseases, and that's basically when your white blood cells just end up attacking your normal body tissue. So these fail-safe mechanisms are very important. So we saw in previous lectures, and we can just harp on it again, T-cell activation is highly dependent on MHC molecules, and these are the major histocompatibility molecules. And so we established that MHC class 1, so there's two types of MHC, it's going to project antigens on all nucleated cells. So therefore, it's not on red blood cells or erythrocytes, because those don't have nuclei. Right? As opposed to MHC class 2, that's going to be on professional antigen presenting cells. So these are going to be your macrophage, for example. That's the name one. And keep in mind, because a macrophage is a nucleated cell, it has MHC1 and MHC2, right? It's not like one or the other. No, like it can have both. But keep in mind, just MHC class 2 is just more for if you're antigen presenting cell, you have that extra one, right? And when you consider what the role of professional antigen presenting cells are, it makes sense that they have an extra way, lots of better words, to present antigens, because that's their freaking job, right? So that's what it is. Again, MHC class 1, CD8 T cells are going to recognize it. MHC class 2, CD4 T cells are going to recognize it. The way you can remember that, MHC class 2, 2, multiple, or 2, right? 4, 2, pair that. And then, or like I want to help, right? So MHC class 2 is the CD4. And then just the other one is MHC class 1 and CD8. Again, CD8 is killer, CD4 is helper. So the T-cell receptor is very interesting structure-wise. So if I start doing a poorly drawn phospholipid bilayer, that won't win me an art award, but it's pragmatic. So that's really good now for what we're trying to do. And it's actually good to be able to contrast this to B-cell receptor antibody. So you're going to notice crossing through phospholipid bilayer, you're going to have like a transmembrane component, but then you have these. So most of the time when we talk about T cell, it's going to be called an alpha beta T cell. And the basis of it being called alpha beta is just a simple fact that it has an alpha chain and a beta chain. So this one that we have here on the left, or right, depending on how you're oriented, is alpha chain, and this is beta chain. And then similar to what we saw in the B cell receptor, there's going to be a variable component and a constant region, right? And so you imagine like the variable part is the one that really varies from individual cell to cell, assuming they're coming from different lineages. And this is what binds the antigen. I believe there's also like a disulfide bond linking them here. So when we say T cell is alpha beta T cell, you're never going to hear alpha beta T cell in a casual you're never going to hear any of this in a casual conversation, immunology conversation, right? If you're alpha beta T cell, you're probably just talking about the default T cell, which is your CD4 and CD8. They fall under this category just on the basis that the molecular structure has an alpha chain and a beta chain, right? Now, in addition to this, I don't think I have this written on the slides, but just know there is signal. The T cell receptor does not work in isolation. It's part of a complex, if you will. So there's a complex, and that is going to constitute of these proteins that flank on the either side. It's called CD3. So CD3, there's two of them, right, one on each side. And you can get specific, but it's like, I think it's like epsilon and then delta. And then there's gamma and then epsilon again. And then there's something called a zeta chain. I already used green, I took another color. And I remember it's just zeta is like a Z, so I just draw like a big Z. It's also transmembrane. So basically, kind of cramped together, right? This is your zeta chain. I'll just put a Z. Your T cell receptor. Oh, and then how can I forget? On the sides. This is going to be either your, depends on the cell. It's going to be your CD4. Wow, I'm atrocious at this drawing. CD4 or 8. Right? Because if it's a helper, it'll be 4. If it's a killer, it'll be 8. So altogether, this is really the bulk of the T cell signaling complex. Antigen will bind the alpha beta T cell receptor, specifically at the variable region. So, like, I don't know if my mouse shows, but we can draw it. It's not okay. So, yeah. So it'll be there. It'll be at the variable region. Okay, and then this will trigger some conformational changes. All in all, this will cause CD3 and zeta chain to do intracellular signaling to, spit out that activation signal needed for the nucleus, or to the nucleus rather. Okay, and then just keep in mind that also part of the binding process, right, is going to be subject to the MHC molecule, and so what makes sure that checks out? CD4 or CD8, right? So again, I drive this home repeatedly. It's MHC class 2 molecule. It's CD4 paired. It's MHC class 1 molecule. It's going to be CD8 paired, right? So helper T cells, CD4, killer T cells, CD8, bind MHC class 1, right? So this is, anytime we talk about a T cell here on out, just assume we're talking an alpha beta T cell, but we'll never call it that. It's because we're going to call it CD4 or CD8, and later we're going to see there's actually subtypes of CD4, which are actually important. It's just not minutia. I assure you that much. But I guess it's subject to the scope of how you want to learn it or why you're learning it. So something to keep in mind, right? But it's here. And then, so let's just backtrack now. So this is the rule. This alpha beta, just ingrained this. This is when you think T cell, you're pretty much going to think this like 99.9% of the time. However, there is such thing as a gamma delta T cell, and its structure is kind of similar. Just instead of naming the two chains as alpha and beta, we call them gamma and delta. Now, these actually don't detect antigen in the same way that the alpha beta T cells do. They're going to depend on something called MICA and MICB receptors. And really, this is still under research, but it's been proposed that these MICA and MICB is just really a stress signal put out by cells, and it's just a come kill me signal. And the gamma delta says, all right, I'll come kill you. So it kind of reminds you like an NK cell in several regards. But keep in mind, NK cell doesn't really look for a stress signal so much as it does just look for an absence of MHC. So if a cell does not project MHC, then case will be like, why don't you project MHC? Everyone projects MHC to some capacity, right? Whether you're doing MHC 1 or 2 doesn't matter as long as you have MHC, right? And so the fact that you don't have MHC means that you're a broken cell, and broken cells tend to be associated with cancer, virus, intracellular bacteria, etc. You will die, or I will kill you, rather. So that's how an NK cell works. Here it's kind of similar in the regard that direct cell killing in some capacity. But there's many mechanisms, and even for the gamma delta T cells, I mean, they're mainly located in the GI tract, and you can think of why that is. It's mainly because, you know, alimentary canal is technically exposed to the outside world. I mean, what are you eating? It's from the outside world, I hope, right? So it makes sense that anything exposed to the outside world has kind of extra layers of protection. And so actually it's kind of controversial to say it, but perhaps you could actually regard gamma delta T cells as T cells of the innate immune system. And I just spoke sacrilege because we talk adaptive immune system. We're just T cells and B cells. Like, that's pretty much it, right? But no, here we actually have these gamma delta T cells who are actually kind of more innate than they are adaptive, right? And yeah, they are. Gamma delta T cells are part of the innate immune system. They're more primitive in the sense that, you know, their mechanisms are completely different than alpha beta. I mean, there's some, you know, there's some similarities, of course, but even the maturation process and the cytokines for different maturation routes and everything, it's distinct. It's distinct, and structurally they're distinct also. So I probably harped on this much more than I should, but that's basically what gamma delta is. You know it exists, but we'll never talk about it again. Here on out, if you hear T cell, we're talking CD4 and CD8, and we have established these are alpha beta, right? And so, in case you haven't been familiar, I've been using these terms already. CD4 and CD8, the two types of alpha beta, right? And so just keep in mind the full name we can use is CD4 alpha beta, but no one's going to say that. You can say CD4. It's just assumed it's alpha beta, but know the signaling complex, or the T cell receptor complex, and also know that this T cell receptor, which I kind of like this. It's like two fingers together. That's what it looks like, and then it just like pokes around with like, where's my antigen? It can, just like a B cell receptor, it can only recognize one antigen, right? Like, really what it is, or to mainly that capacity, right? And it can only bind one antigen also. So this is opposed to the B cell receptor, because it has those two FAB regions, one on each arm, one like monomer of immunoglobulin can bind two epitopes, right, on antigen. Suppose T cell receptor only binds one. Something else you can also make a distinguisher. B cell receptor is antibody, and then, you know, antibodies, well, really it's antibody tethered to B cell surface, but antibodies are not necessarily just bound to B cell surface. In fact, they can be circulating in the plasma, right? As opposed to T cell receptor, there's no such thing as a circulating T cell receptor. It's just fixed to the T cell surface. Okay, and so really the way I think about it, like I said, it's like these two fingers, because alpha beta, right, and just prodding around on those whatever MHCs it can find in its journey in life. Okay, so that's what we're doing here. So I think I can clear. Nice. Getting better at the Zoom thing. Sorry. It's only been 2 years, right? Okay. Oh, I had to include this. There's also such thing as a natural killer T cell. So this is also a type of alpha beta T cell. However, it's not as diverse. So very similar to, you know, like the B cell receptor. You know, we have receptors for antigen that theoretically don't exist. Name of the game. Same thing for T cell receptor. Even the enzymes they use are the same. It's RAG1, RAG2, you know, just for like making the making the receptor that can bind antigen, right? Natural killer T cell, yes, it's also alpha beta in nature. So it's not like the gamma delta, right? However, it's less diverse. And so it's not as, and it's still something under study. And we'll never talk about it after this. But basically, there is a surface marker called CD1, and it's kind of acts like MHC1, right? But instead of projecting protein antigens, which I think we established before in previous videos, the MHCs are going to project, peptide antigens. CD1 is actually going to project lipid antigen. Such things exist. And so it's like MHC1-like, and I guess it's fortunate that CD1 looks like MHC1. But basically, a natural killer T cell will do antigen detection on these, right? So yes, we have lymphocytes who can recognize non-peptide antigen, but they're not the mainstay discussion. Here on out, if we're going to talk about T cells, we're talking about alpha beta T cells, CD4 or CD8. That's it. So we'll never see gamma delta again. We'll never see natural killer T cells again, but they exist. That's the point. So T cell receptor signaling, we established this before. The alpha beta, the true receptor, we have the variable region. It'll bind antigen on respective MHC or respective MHC where it's found, like wherever the antigen it is, right? And the rest is CD3, and also so we established the zeta chain. Hey, and we can make a connection here. You know how CD3 is a signaling molecule we established as part of the complex? Where have we seen a similar trend before? Ig alpha and Ig beta of the B cell receptor, and they do intracellular signaling. So their CD3 and Ig alpha and Ig beta are similar in that regard. And even like for the CD3, right? CD3 has like the epsilon delta or epsilon gamma, right? So it's kind of like there's like two of them. Just like there's Ig alpha and Ig beta, right? So the more you know. So yeah, and again, and we established this before, and we're just going to keep harping on it. Antigen on MHC, right? So the MHC holds the antigen from whatever cell in the body, so I don't know, endothelial cell. So it's not a professional antigen presenting cell, so it's going to have MHC one, yes. So MHC1, so it's going to project antigen, and then a T cell receptor will come along, with a T cell receptor will come along, and it will bind that. And then because it's on MHC class 1, for anything to happen, CD8 on that T cell has to bind to the MHC1, and that's how you can have an interaction. But the T cell receptor to the antigen on the MHC itself is not sufficient. You also need that. You also need that CD8 or CD4 subject to the cell type it was discovered on, or antigen where the antigen was discovered on, to stabilize that bond, right? And this bond stabilizer, whether it's CD4 or CD8, is what gives the T cell its name, and that's a very important distinction to make. Antigen presenting cells also project B7, which binds to T cell CD28, and this is an added layer of co-stimulation. We discussed this in the last presentation, but like I said, this presentation just builds on everything. I have a slide toward the end of this presentation that puts everything together, and there's also an added step. So but it's give you the full picture. That's what we want. So really, when I'm talking about right now the T cell activation, we kind of fast forward in the timeline of the life of the T cell, because really before T cell activation comes T cell maturation. And so it's funny, like in pretty much every immunology course I've come across, when it's taught, T cell activation is taught before T cell maturation, even though T cell maturation happens first. So here we're actually rewinding behind activation. Now we're at the maturation phase. So any white blood cell is going to have its start in the bone marrow. It's pretty much any blood cell, right? However, unlike the B cells, the T cells will make their way over to the thymus, right? And your thymus is an organ in your neck area that actually involutes with age. So if you look at like a 50-year-old man, probably the thymus is well on its way out, if not already out the door. So, but that's normal. The thymus involutes with age, but it exists. But the thymus is important because it's actually the T cell maturation area. Okay. So T cells, when they're like very primitive, very early on, when they're babies, lots of better words, they actually start off as double positive. They have CD4 and CD8. In the thymus, we have a whole set of processes that will basically make the T cell commit to either being a CD4 lineage or CD8 lineage. And really, that happens based on where does that T cell find its cognate antigen first? And you can be like, where does it find its cognate antigen first? What do you mean? We're in the thymus. How are you going to find, I don't know, viral or, I don't know, not really viral, sorry. How are you going to find, because here we're testing for self-tolerance. If you're in the thymus, right, and you're trying to make sure that your cell is not self-reactive before it can go onto the system, how can you ensure that the thymus is going to represent kidney antigens, for example? And that's because the thymus has like this autoimmune regulator type protein called AIR. And AIR actually is like a simulator of the body. So the thymic cells can actually project renal-like antigens. And if the T cell can detect them, it's like, okay, that means you're self-reactive. That means you cannot go on, right? But there's like a whole lecture on this process, which is quite amazing. But what's just the takeaway here? During this, like the simulator, if you will, if it finds this cognate antigen on MHC1 in the thymus or MHC2 in the thymus, whichever one it finds first, that'll determine if it's going to be CD4, CD8. So if it finds its simulator antigen, if you will, on MHC class 1, the double positive, then we'll want to commit to being CD8 because it used its CD8 before it used its CD4. But if it used its CD8, well, that's it. Okay, it's going to upregulate CD8, downregulate CD4, and then it will go on in its lineage to mature into a CD8 cell. Conversely, if it found its cognate antigen on MHC class 2, that means it used its CD4 first. And if it uses CD4 first, it's going to stick to being CD4 the rest of its life. Vast oversimplification, but I think it works for today. I'm probably already over time. And then this is just explaining what I just said. Yeah. Okay, so let's talk about the full picture now. Okay, so we're out of the thymus. So whatever, you have a mature T cell. It's naive though, right? A naive helper, naive simply meaning it hasn't met its match in the circulation and hasn't become an effector. Okay, so it's kind of like how do I put this? So it's kind of like it's fresh out of medical school, naive, right? I mean, yes, has its license, and yes, it can do stuff, or it has the propensity to do stuff, but it's not seasoned yet. Hasn't been activated. It hasn't become an effector, right? Like just because you have like the degree doesn't mean you have necessarily like the know-how yet, right? So yes, but all the foundations are there. Right? That's what it means to be naive, right? So then once it finds its match in the circulation, like around the body, in the lymphatics, then it will become a effector cell, which is a functioning cell, right? Which if a CD8 becomes effector, that means it's a killing cell. If a CD4 becomes an effector, that means it's a cytokine factory. That's what that means. So in a lymph node, we have a naive helper T cell. It finds cognate antigen on MHC class 2. Good. And that's actually going to be on a dendritic cell. Yes. Because dendritic cell is one of the professional antigen presenting cells. Recall from like previous lecture, the dendritic cells like the tattletale. It uses TLRs, picks up a danger signal. It's going to gulp up all the antigen in this area, and it's going to swim to a lymph node. That swim takes about a day. And then it's going to take up shop in the paracortex of a lymph node, and it's going to have its MHC class 2 projection and MHC class 1 projection of the antigens it took up when it would gulp up everything in the periphery, right? We're in the paracortex, and MHC class 2 has the antigen that we're looking for. The respective CD4 cell finds it, okay? Now, this step 3 right here, I think we're introducing for the first time, which is fine, but we're introducing it now, okay? I like to think of CD40L like a finger, because the L is like the finger, right? kind of. Not a gun, more so a finger, right? So CD40L is on the T cell. So the T cell has a finger, right? Or many fingers, I don't know. And it's going to bind to CD40. So CD40L just means ligand. So make sure so it makes sense the ligand binds to CD40, right? CD40L binds to CD40, right? Yeah. When this happens, you can kind of think of it as like the prerequisite on button. When CD40L presses CD40 on the dendritic cell, so I think it's like a finger pressing the zero is like a, right? So this is going to cause upregulation of co-stimulatory molecules needed to make sure that the mainstay, like the bread and butter of the interaction, can proceed. And what are the co-stimulatory signals? So we discussed these already. It's going to be CD8 on the T cell, and it's going to be binding to B7 on the dendritic cell. And again, remember, B7 can be also called a CD80, CD86 as well, alternate names for the same thing, but we're saying B7 here. So again, CD40, CD40L is like the permissive button that will allow co-stimulatory markers to get upregulated. What are these co-stimulatory markers? On the T cell, it's going to be CD28. And on the dendritic cell, here in this case, it's going to be B7. And these are what they need. So again, we draw a picture, mental picture. We're drawing a T cell surface. What's on the T cell surface so far? T cell receptor, CD3, zeta chain, that whole complex. CD4 or CD8 subject. Here we're talking about CD4 because that's going to stabilize the bond. What else? CD40L, because that's like the button presser. And then what's on the dendritic cell, for example? MHC class 2 holding the antigen, right? And then because it's CD4, that's why I said MHC class 2. CD40, because that's what's going to get pressed by CD40L on the T cell. And yeah, so that's if we're just drawing out that picture. So CD40L binds CD40. Okay, this causes co-stimulatory markers to get upregulated. Now, T cell receptor is going to bind its cognate antigen on the MHC, right? Here, because again, it's helper T cell and dendritic cell, a professional antigen presenting cell with its, right? That's because helper T cell, it's CD4, and it's going to bind MHC class 2 on the dendritic cell, right? That happens. Then you're going to have CD4 on the T cell, stabilizing that bond between the T cell receptor and the antigen, right? You already have CD40L on T cell binding to CD40 on the dendritic cell, okay? This signal can't proceed though because we just need one more. And what is that one more? CD28 on the T cell to B7 on the dendritic cell. Once you got all these, and I know it's a lot, then it's like the clear signal. It's like, okay, you can become an effector now, right? Upon activation also, IL-2 receptor on T cell can get upregulated, and IL-2 receptor binds IL-2, and IL-2 is just simply a T cell growth factor, and I think it's very fortunate that 2 and T cell both start with T. And there's other T cell growth factors subject to specific subtype. I think like IL-4 can be a T cell growth factor, but it's for a specific subtype of helper T cell, but that's not the topic of today. Okay. So that's what it is. You get an activated T cell, it becomes an effector. That was the whole point. So again, what's the point of having this be so complex, be so multi-step, have all these checkpoints? Because again, if you're activating a T cell, it's a big deal. Big deal. So we have to clear. I'll figure it out. Okay, good. Clear all drawings. Okay, so we just did give an example of a CD4 cell. CD8 would be very similar, except it would be MHC1. Now, mechanism slightly different for the CD8 cell aside from MHC1 in that it can occur by two means. And we're going to get a little bit passionate here, if you will. There's a 3-cell mechanism which activates CD8 T cell, and there's a 2-cell mechanism. Okay, I'm going to start with the 3-cell mechanism. It's the better of the two because it takes more work for more people. I don't know what is that social loafing of a thing, but not really in the world of white blood cells. And I hope not. So 3 cells and the 3-cell mechanism of CD8 activation. What is it? Okay. So 3 cells are going to meet in the lymph node, again in the paracortex. It's going to be a naive CD8 T cell. Okay, so fresh out of the thymus. A dendritic cell with its cognate antigen on MHC class 1. Now keep in mind, the dendritic cell also does have MHC class 2, yes. But in the context of CD8, what does CD8 recognize, MHC 1 or 2? 1, okay. So yes, the dendritic cell can do both because it's nucleated, so it has 1, and it's a professional antigen, so it has MHC class 2 as well. But in the context of CD8, we only care about MHC 1. Yes. So naive CD8 cell, dendritic cell with cognate antigen MHC class 1, and also helper T cell. And you're going to be like, but if it's a helper T cell, I mean, is it getting activated also? Possibly, but in the context of activating a CD8 cell, this helper T cell is already an effector, really. It's helping out. That's the name. So yes, it can act as a cytokine factory in the systemic circulation and whatnot, but also within the lymph node, it can facilitate CD8 maturation or activation, rather. Not maturation. We already did maturation. Activation. Okay. So what's the deal with this, though? To have this 3-cell mechanism, why is it a bigger deal? And why is it, I mentioned it, why is it later? That's because to have an effector CD4 cell, right? That means you already geared up your adaptive immune system, and you went through all the processes that we went through on the past slide, what it takes to activate a CD4 cell. So that means to have a CD4 cell available to facilitate an interaction between a naive CD8 and a dendritic cell, you've already been fighting that infection. Your immune system fight has already been on for some time, right? So the 3-cell mechanism is not an immediate relief. This 3-cell mechanism can only occur later in an infection because it requires mature, activated effector CD4 cells, right? So this is why the 3-cell mechanism works better. Now, what's the benefit of the 3-cell mechanism? The T-cell, the CD8 T-cell product from this is better, if you will, lots of better words. And so we're going to talk about what that means after we talk about this 2-cell mechanism. So 2-cell mechanism, same setup, but this occurs early in an infection. So what do you think the difference might be? If I said the difference for 3-cell mechanism is that you had these effector CD4 cells who can facilitate the interaction and help with the 3-cell mechanism, how might a 2-cell mechanism be different? Well, if you're thinking that we're going to take away these helper CD4 cells because they're not available yet, you would be correct. So this 2-cell mechanism, you have a naive CD8 T cell and a dendritic cell in the lymph node who meet. Now the dendritic cell is going to take over the facilitation job of the helper T cell. So the dendritic cell really in this relationship wear the pants. I don't know why it does, but it does, and we're thankful that the T cell does that. I don't know, perhaps they have a story of unreciprocated love, and the dendritic cell really needs its niche in life. Not really necessarily I'm condoning that, but, props to the dendritic cell to take on that burden. I wish it didn't have to, but it does. But help comes because it gets better one day, right? Or at least we tell ourselves that until it does. And then we look back at things and like we got through it, right? What am I saying? Anyways, so you have this dendritic cell. It's going to do facilitation and antigen presentation. And that facilitation job is formally or would belong normally to CD4, but those aren't available yet because it's early in infection. So what gives? Well, really, you're going to compromise quality. So you had 3 people working on it, and I have 2 people working on it, and the quality goes down of the CD8 cell. So the CD8 effector cells produced from a 2-cell activation mechanism, these are actually more short-term. So they can't become memory T cells, and they tend to be more short-lived, so they don't have as much longevity. Like they'll tire out and die faster, as opposed to from the 3-cell mechanism, you're making quality product. But we can't, flame this 2-cell mechanism. It's very important because it's pretty much the only mechanism we have running until CD4 can come in and facilitate the 3-cell mechanism. So we have to celebrate the 2-cell mechanism because early in the infection is the only thing we have, or I say the only, but really it's the main thing we have running for us. So the short-term but immediate T-cell systems at the start of an infection will hold off the fight until the 3-cell mechanism can kick in once those CD4, those effector CD4s can roll in and aid with facilitation. So now at the conclusion of this PowerPoint or lecture, or both, right? You activated the T cell. If it's CD8, what does it mean to become an effector now? So it will upregulate its IL-2, which we established, IL-2 receptors, which we established the T cell growth factor. This will make it more receptive to proliferation, making clones of itself. It will enter the circulation in search of MHC class 1 molecules on all nucleated cells, displaying that cognate antigen that it initially encountered on the dendritic cell in the paracortex of the lymph node. So it discovered what it target identified, if you will, is what it learns in the lymph node, and then it goes out into the periphery to find that target and kill it. So it's quite intense. So again, target identified in the paracortex by the great teacher, the dendritic cell, or in the case of the 2-cell mechanism, it's not really a great teacher, it's great teacher and the one who wear the pants and carry the relationship. I'm not going to call it one-sided, but it makes you wonder, right? What am I saying? Anyways, so what does it mean to become an effector CD8 cell? I established already that it can engage in direct cell killing, much like an NK cell. What does that mechanism do? CD8 T cells can employ something called perforin, and basically it perforates the membranes of infected cells and even bacteria to some extent, right? And then after so it pokes a hole in the pathogen, and then in that hole it will dump poison, and that poison is cytotoxic granules, and some of that's like granzymes and whatnot. And these granzymes and whatnot will trigger apoptotic cascades within infected cell or bacteria. CD8 cells can also release tumor necrosis factor and interferon gamma, and all these amplify the macrophage response. And the macrophage, is like this very vicious big eater cell, and having them on your side is great in controlling an infection. And mind you, because there's always macrophages, they're part of an innate immune system, they're always on, they're always there. But then once you hyperactivate them, then the real battle's on, and they get significantly better at what they do, right? And the list goes on. And then once you activate an effector CD4 cell, or once you have an effector CD4 cell, what does it do? Same thing, it will upregulate its IL-2 receptors, so it can, be pro, amplification type phase, right? Self-stimulates, if you consider that actually some CD4 cells actually produce IL-2, and then they have IL-2 receptors, so it self-stimulates to make even more of itself. That's cool, but we'll talk about that later. But generally, the CD4s are going to be cytokine factories. They're going to direct immune response, but also they can assist dendritic cells in the lymph nodes for the 3-cell mechanism to make more quality product CD8 cells, right? So that's what that is. And I hope I didn't burn anyone out. But that's pretty much it for activating T cells. And again, we'll touch on these subjects. Really, it's my intention with these. I try to touch on subjects again and again. You're probably getting sick and tired of me saying, Oh, look, if anytime I say CD8, I was like, oh, but by the way, CD8 is MHC class 1 and CD8 is killer T cell and da 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 da. Like, yeah, I know I repeat a lot, but that's what I guess TOPE is like, just consolidate, because all these concepts keep coming up again and again and again. But next time, we're actually going to talk about functions of mature T cells. So that last slide when I said what each one does, it's great. But when we talk about the CD4s and specific effector CD4, there is subtypes. And so there's a whole talk on the subtypes of CD4 cells. These are my sources. I'm right now in an inflamed appendix, and this has lymphocytic infiltrates, which can be B cells and T cells, since I felt appropriate for the background. Yes, so these are my sources. Thank you all. I hope you're all doing well. And please let me know if there's any topic you'd like me to discuss. For now, I'm just trying to chug through this general immunology, and then we'll get to more specialized topics in immunology and microbiology and whatnot. So I hope it's helping you. I mean, that's the whole point. And yeah, just let me know if there's any topic you'd like me to discuss in specific in the future. Please, I appreciate that. So everyone, take care, and I will see you next time when we talk about specific CD4 subtypes. All right, take care.